Hello, my name is Seema Agarwal, and I am an associate professor at the Center for Cell Reprogramming and in the Department of Pathology at Georgetown University. My laboratory is focused on establishing and studying patient-derived models of metastasis and was recently awarded $100,000 by the Ruesch Center for Research on GI Tumors starting from July of 2021. Today, I would like to express my thanks for the research funding and briefly describe our research and how this funding is helping to translate our research into technologies that will ultimately improve the lives of patients with cancers that are some of the most difficult to treat. Metastasis is an intractable clinical problem. Most cancer deaths, in fact 90% of them, are related due to the metastasis. This makes metastasis is the main driver of cancer death. Unfortunately, currently there are no approved drugs to target metastatic cells. The primary reason for this is the poor understanding of the metastatic process, and no druggable targets are identified or validated for metastasis. Just to give you an overview of the metastatic process, from the primary tumor node, some of the cells escape the primary tumor node and intravasate into the nearby blood vessels. And these tumor cells that circulate in the blood vessels are called circulating tumor cells. And this is the focus of my research. After a while, some of the cells of circulating tumor cells extravasate into the nearby organ to create the metastasis or the spreading of the tumor in patient. In 2008, the FDA approved Cell Search System, an antibody-dependent method for the isolation and enumeration of circulating tumor cells from a patient's blood sample. I will continue to call circulating tumor cells as CTCs in the rest of my talk. Using this system, it has been shown in the pancreatic cancer patients that even if in the blood samples, if one cells can be identified, that puts the patient at the high risk for the overall survival, compared to the patient blood samples which has no identifiable circulating tumor cells, and they had the much better overall survival and prognosis. And similar results were obtained in 68 patients of colorectal cancer patients, where any identifiable circulating tumor cells led to the poor prognosis and shorter overall survival compared to the patient samples where there was no identifiable circulating tumor cells. So this makes the circulating tumor cells as the key player for spreading the tumors in patient, and we should study this. So if counting the circulating tumor cells in an individual patients predict the clinical outcome, and if we can use the advanced genetic and proteomics and bioinformatics approaches on circulating tumor cells, then we can lead to the personalized patient care and discovering new targets for metastasis. However, to do so, we need to have a sufficient number of CTCs from each patient's blood. And that is a very big challenge currently in the research field. And why is that challenge? Because these circulating tumor cells are very rare in the blood. They are less than 1 in 100,000 of other blood cell types. They are fragile and they are short-lived. Thus, it is very difficult to accurately identify a representative population of circulating tumor cells in the blood samples from a patient. And it has become necessary to apply the genomic and proteomics tools that we need to expand these in the cultures and to further expand these from the culture to generate the xenograft model so that we can do a personalized medicine approach. And this is what we bring to the table. Last year, we published a paper in which we have shown that in a very simple protocol, we can use the patient blood samples and can grow the circulating tumor cells in a very high success rate. And the key features of this method, which is under patent right now, is the unbiased selection of circulating tumor cells. So no antibody and no size-based selections, and no harsh method to isolate circulating tumor cells from blood samples. So in the right shows the table compared to the other methods that have been published prior to ours. Their success rate is much lower than our success rate, where we used 12 breast cancer patient samples, and we were able to create a short-term circulating tumor cells cultures from all these patients. Since then, and with the help of Ruesch Center funding, we have expanded this to different tumor types. And what I am showing here is the colorectal cancer, which has a very poor survival rate if metastatic setting. And so does the liver and pancreas, which have even lower survival rate when the cancer spreads and metastasizes. And what is shown here, with the help of Ruesch Center funding, we are able to create short-term cultures from cholangiocarcinoma, from pancreatic and colorectal with a very high success rate. I'm sorry it is not showing in the table, but we had about 85% success rate with all tumor types that we have tried so far. Not even that. I just told you that we have made a short-term circulating tumor cells cultures, but that is not enough, because we want to have these cells available to other researchers to use it for more assays and for personalized medicine approach. So to expand it for a long term, we used the culture circulating tumor cells and injected into immunocompromised mice, and we were able to create the tumors from breast, colon, lung, pancreatic, and very recently from cholangiocarcinoma blood samples. And our tumor or our xenograft models or mouse models have been made in less than 4 months compared to the others which have taken at least 6 to 12 months to create these mouse models. And this is really important that we are able to create this in less than 4 months, because then there may be a window for us to do a personalized medicine approach. So this slide shows our collaboration with Ruesch Center clinicians. We work with them very, very closely. With Dr. Ruth He, we are in the process of collecting 15 cholangiocarcinoma. We have already collected 12. It is a very rare and aggressive cancer types. And we have established not only the short-term CTC cultures, but use these cultures to create the CDX models, which are the mouse models. For further molecular characterization. With Dr. Marshall and Dr. Weinberg, our aim is to collect 20 colorectal cancer patients and create the circulating tumor cells and CDX mouse models. With Dr. Marcus Knoll, our goal is to collect pancreatic cancer patients and to do the similar things to create the CTC and CDX model, and then apply these models that we create to exhaustive genetic and molecular characterization and compare it with the parental tumor to identify the markers for drug and as a biomarker to predict which patients will be at high risk and which patients will be at the lower risk for survival. Leveraging the Ruesch Center grant, the following papers and grant applications are based on work initiated with the clinicians from the Ruesch Center. I have given you just the 3 papers and 3 grants that I envision by the end of this year and early next year. First paper would be in using the cholangiocarcinoma models in their genomic analysis, and we hope to apply for NIH, DoD, and or foundation grants by early 2022. Paper 2 and grant 2 will be on pancreatic cancer models, since we have already established 11 circulating tumor cells from pancreatic cancer patients and established already 5 of CDX models. And so we hope that in sometime in the middle of 2022, we should be able to submit a paper on pancreatic cancer and apply for the grant as listed here. And the third paper next year, which we envision will be from colorectal cancer patients using the same techniques and technologies as with the cholangiocarcinoma and pancreatic cancer patients. And each grant will be more than $1 million and expand over 3 to 5 years, depending on whether it is an NIH grant or Department of Defense grant. Before I end, I would like to bring the attention of how this funding is helping me to train 2 physician scientists in the lab. One is Jerry Xiao. He's a very intelligent and hardworking MD PhD student in my lab, and he is the one who spearheaded this project and already has published 1 paper on breast cancer, and then he's ready to publish another paper soon on circulating tumor cells. And his goal is to finish his PhD in February of 2022 to get back to the medical school and start his 3rd and 4th year of medical school starting from March 2022. Another person in my lab is Shravanti Suguru. She has a medical degree from India, and she is interested in learning lab techniques and apply her medical knowledge to projects with translational and clinical application, such as the circulating tumor cell project in my lab. And she plans to start her PhD in 2024. So I'm very excited to train these 2 physician scientists, which will do a very good job as they move forward from my lab. And then I would like to thank 2 of my co-inventors for the patent on circulating tumor cells. One is Dr. Richard Schlegel, who is a director and founder of Center for Cell Reprogramming, and Paula Pullman, who's a oncologist in Georgetown, and recently she has moved to MD Anderson Cancer Center. These 2 are co-inventors with me on the patent. First and foremost, acknowledgement to the patients who consented and provided blood samples. The SRBSR core facility at Georgetown, and specifically to Colleen and Samira, who are diligently consenting patients and collecting blood samples for this project. Ritu Mukherjee, who is currently a GI fellow, and Laura Coleman are a key to identify patients whose samples we can collect under the IRB protocol. And my collaborators on bioinformatics, Hani Godarji from University of California, San Francisco, and Walzamo and Agnastu from Johns Hopkins University. Without their help, this project would not be possible. And I would like to thank and leave you with the last slide, which really brings on home how the Ruesch Center grant is going to help the patients and basic science and translational science. Thank you.